Order! Order! You are an incorrigible delinquent at times. Behave yourself, man!
There is new hope tonight for thousands of families who live with the prospect of passing on inherited diseases to future generations. For the first time, scientists have successfully repaired a faulty gene in human embryos. The US and South Korean team used a process known as gene editing to correct DNA that causes a deadly heart condition. The controversial technique is still at the early stage, but it raises the hope of preventing 10,000 gene disorders which pass down the generations. Our medical correspondent, Fergus Walsh, reports.
The goal could not be more ambitious: to eradicate inherited diseases. These scientists have taken an impressive first step on a long road, editing DNA in human embryos. So how was it done? Inside the nucleus of each of our cells is our genome, billions of pieces of DNA. It's the instruction manual for life. The scientists were targeting a faulty gene that causes a serious heart condition. They fertilized a healthy egg with sperm from a man carrying the faulty gene. They then injected the gene editing system known as CRISPR. This scans the DNA like a spell check or a sat-nav. It then cuts both strands of the DNA and removes the faulty gene. A healthy copy of the gene from the egg was then naturally inserted. Now here are some of the embryos from the study in the journal Nature after being edited. 42 of 58 embryos were corrected. They were allowed to develop for 5 days. None was implanted.
We're very excited about all the work, of course.
The research has been welcomed by a team in London who have a license to edit human embryos. They say the technology could eventually help many families.
There are some nasty genetic diseases, such as Huntington's, or as in this case, a disease that affects heart function later in life, which can basically blight families for many generations. So a method of being able to avoid having affected children passing on the defective gene could be really very important for those families.
Nicole Mowbray has the same heart condition which was corrected in human embryos. She now has a defibrillator implanted in her chest in case her heart stops. She has a 50% risk of passing on the condition, but is unsure whether she'd ever consider gene editing.
I wouldn't want to pass on something that caused my child to have a limited life or a painful life or a life of risk. I mean, that does obviously come to the front of my mind when I think about having children. I wouldn't want to create the perfect, in inverted commas, child. I feel like my condition makes me me.
Previous attempts at editing human embryos in China led to serious errors in the DNA, so there's a lot of work needed before this could be considered safe. And it raises ethical issues about how far science should go to create healthy babies.
And Fergus is with me now. Fergus, a big advance, as you suggested, but let's be clear, this is not something that's going to happen next year.
Absolutely. We're at the foothills of this technology. There's still a mountain to climb. And this is controversial, George. There are moral questions here. Critics will say this is the road towards designer babies. But let's be clear, what we're talking about is designing out faulty genes that cause terrible inherited disorders, often fatal, not designing in positive traits. But critics will say that will come next. Now, couples at risk of passing on a genetic disease already have options. They can have embryo screening through IVF, but this will allow more healthy embryos to be selected for them. But let's be clear, we are years away from potentially curing genetic disorders using gene editing, and still further from enhancing humans.
Fergus, thank you.
To a big question now. Imagine a world with no inherited diseases. Well, impossible as that sounds, it could be British scientists who are tasked with making it happen. Their colleagues in America have just altered the DNA of an embryo with faulty genes, but they're restricted on taking it to the next stage by ethical regulations on that side of the Atlantic. Now they're hoping scientists here will take up the challenge.
The genetics revolution is giving us a profound understanding of how our bodies work and how things can go wrong. Where they do go wrong, scientists can try to fix them by precisely editing a person's genome using a new technique called CRISPR. Today, in a landmark study, scientists showed that they could use CRISPR to safely remove harmful genetic mutations in human embryos.
At the moment, we can check embryos to look at some aspects of their genome. Using this procedure, genome editing, we could actually go into the abnormal embryos and change them and correct them so that they wouldn't carry that disease. So that really, scientifically, is an amazing step for us, and it really is opening up so many possibilities for the future.
In today's research, scientists use CRISPR to correct an abnormal gene that causes a type of sudden heart failure. The scientists fertilized a healthy egg so that the embryo contained one mutated gene and one normal copy of the gene behind the heart disease. They then used the CRISPR technique to insert an enzyme called Cas9, which made a cut on the mutant gene sequence. The embryo subsequently repaired the cut by itself, using the healthy gene from the egg as its template. The process was remarkably efficient. 2/3 of the embryos created in the experiment ended up with 2 normal copies of the gene. The research is only a proof of concept right now, but it has huge promise. CRISPR is one of the most exciting tools in genetics. Scientists around the world, including here at the Francis Crick Institute in London, are working to make it safer and more efficient in the hope that one day it'll turn up in our clinics, where doctors can then use it to treat the thousands of inherited diseases that have so far been beyond the reach of medical science. CRISPR could be used for a wide range of genetic conditions, from cystic fibrosis to certain types of breast cancer. But scientists caution against going beyond necessary clinical applications.
It's important that the first attempts, at least, are to do with serious genetic diseases. And no one contemplates doing it for more trivial things, or for so-called designer babies, or trying to do enhancements. That would be very foolish. It's absolutely not worth the risk.
Clinical uses for CRISPR in humans are years away. When they come, they'll usher in a new medical world. Alok Jha, News at 10.